Provide the summary of the video script in 33 words. Welcome to the lecture on enzyme kinetics and the Michaelis Menten equation. We will explore enzyme-substrate interactions, factors affecting reaction rates, mathematical equations, and graphical representations. We will also discuss enzyme inhibitors, allosteric regulation, drug development, advanced concepts, and key takeaways. Let's get started. The Michaelis Menten equation is a mathematical model that explains how the rate at which an enzyme converts substrate to product depends on substrate concentration. It introduces the Michaelis constant, Km, representing substrate concentration at which the enzyme is working at half its maximum velocity. This knowledge is important for understanding enzyme regulation and developing targeted therapies. Enzymatic reaction rates measure how quickly enzymes convert substrates into products. Several factors influence reaction rates, including substrate concentration, enzyme concentration, pH, temperature, and the presence of cofactors or coenzymes. The reaction rate is typically determined by the frequency of successful enzyme-substrate collisions and the speed at which the enzyme converts the substrate. At low substrate concentrations, the reaction rate increases as substrate concentration increases since there are more opportunities for collisions. However, at higher substrate concentrations, the reaction rate reaches a maximum, known as the maximum velocity or Vmax. The Michaelis Menten equation is a fundamental tool for studying how enzymes work. It shows how the rate of a reaction is influenced by the concentration of substrate molecules. By analyzing experimental data, we can determine important parameters like Km and Vmax. The Lineweaver-Burk plot and the Eadie-Hofstee plot are commonly used graphical representations in enzyme kinetics. These plots help determine Km and Vmax by linearizing the Michaelis Menten equation. Determining Km and Vmax is key to studying enzyme kinetics. One approach is measuring initial rates at different substrate concentrations, plotting data on a Lineweaver-Burk plot. Alternatively, rearranging the Michaelis Menten equation yields Km, revealing substrate concentration at half of Vmax. Precise determination helps assess enzyme efficiency, substrate affinity, and limitations in physiology or disease. Enzyme inhibitors are molecules that hinder the activity of enzymes. They can be reversible or irreversible, and can either compete with the substrate or bind to alternative sites on the enzyme. Understanding enzyme inhibition kinetics is crucial for drug development. Allosteric regulation plays a significant role in enzyme kinetics. Allosteric enzymes contain regulatory sites, distinct from their active sites, that can bind effector molecules. Binding modulates the enzyme's activity, either enhancing or inhibiting it. Allosteric regulation can be positive, activation, or negative, inhibition. Positive regulation occurs when the binding of an effector molecule enhances the enzyme's activity. Negative regulation, on the other hand, occurs when the effector molecule inhibits the enzyme's activity. Allosteric regulation allows for fine-tuned control of metabolic processes and is essential in maintaining homeostasis. In summary, enzymes can be regulated through post-translational modifications, enzyme synthesis regulation, and reversible covalent modifications, which provide a rapid response to cues or pathways. Understanding these mechanisms is crucial for comprehending enzyme kinetics. Enzyme kinetics plays a significant role in drug development. Understanding an enzyme's kinetics can aid in designing drugs that specifically target enzymes involved in disease pathways. In drug development, Km values and inhibitor constants, key, are crucial parameters to consider. High-affinity drugs typically exhibit low Km and key values, indicating strong substrate or inhibitor binding. Furthermore, enzyme kinetics assists in determining optimal drug dosages, predicting drug-drug interactions, and evaluating potential adverse effects. By studying the kinetics of enzymes targeted by drugs, scientists can optimize drug therapies and increase their effectiveness while minimizing side effects. In this section, we will explore irreversible enzyme inhibition kinetics, focusing on the formation of covalent bonds by irreversible inhibitors. This process helps us understand enzyme structure, specificity, and potential drug targets. To analyze irreversible inhibition kinetics, we use pseudo-first-order kinetics, assuming a higher concentration of the inhibitor than the enzyme. This helps determine the reaction rate dependent on the concentration of active enzyme. Important constants like the second-order rate constant, K2, and the dissociation constant, K, are determined through kinetic analysis. Structural analysis techniques, such as X-ray crystallography and cryo-electron microscopy, are employed to study enzyme-substrate complexes at an atomic level. Determining the three-dimensional structure of enzyme-substrate complexes provides crucial insights into substrate binding modes, active site architecture, and catalytic mechanisms. Visualizing enzyme-substrate interactions at a molecular level helps in understanding the relationship between enzyme structure and function. It also aids in the design of specific inhibitors or modulators targeting enzymes involved in disease pathways. The Michaelis Menten equation is limited in predicting reaction rates when enzyme concentration is high. It also assumes simple kinetics and one binding site, while many enzymes have more complex kinetics and multiple binding sites. Advanced models consider these factors for a deeper understanding of enzyme kinetics in biological systems. In the conclusion, we covered enzyme kinetics, including enzyme-substrate interactions and more advanced math models than the Michaelis Menten equation. We explored topics like reaction rates, Km and Vmax discovery, enzyme inhibitors, and enzyme kinetics in drug development. Understanding enzyme kinetics is vital for comprehending the intricate mechanisms of enzyme function and their significance in physiology and disease.